Good afternoon, student. Today I am going to teach you about the phylogenetics or how to make a phylogenetic tree. What is phylogenetics? What are different types of method which can be used for phylogenetics? Actually, phylogenetics is very important to study in bioinformatics because it tells you the evolutionary relationship between the different species, different organisms, how they have been evolved during the process of evolution. So this is very important. In the Coursera course, the week 5 also describes about the phylogenetics. If you have read that week 5 course material as well as seen the video. However, it's not a problem because I'm going to explain you that in a simpler way, and we'll also do an exercise for making how to make the phylogenetic tree with the help of Mega software. So initially, first we'll read about what is phylogenetics, how to make the tree, and then we'll go to the real exercise. All right, so I'll share our presentation with you right now. Yeah, so this is a presentation. We'll read. Yes. So, yeah. So this thing which we are seeing here is that how different species are connected to each other. So this is kind of a, you know, a phylogenetic tree, which is showing the relationship of one organism with another. This is mostly a, showing the phylogenetics between the higher organisms. So if we go back to the time, we'll see that the Charles Darwin was a person who has actually started the phylogenetics, right? Because he started trying to find the relationship between the different species, different finches. And in that way, he was a person who had evolved the genealogical trees of different species of which are found in the nature. So in his book in Origin of Species, there's a sentence which was written: "The time will come, I believe, though I shall not live to see it, when we shall have fairly true genealogical trees of each great kingdom of nature." So that was the sentence. So he believed that maybe he will not live at that time, but there will be a time come when we'll have a true genealogical tree that we'll know at which species born at what time, evaluation of which species came was done at what time, and how all these species are related to each other, right? So and what is molecular phylogenetic? Molecular phylogenetic is a study uh, of relationship between the species on the basis of its molecular structure, on the basis of its DNA. Okay, so similarly, an example which is given here is that there are different species which are written here, and if we want to see the relationship between them, we'll find that the crocodiles, this kind of tree, that the crocodiles are birds are together, then the lizards and snakes are together, rodents and primates are together, and then in this same group marsupials are there, which shows that if you see in this tree that all these reptiles and aves actually are belonging to the same clade, where the lizard and snakes are more similar to each other, and then crocodile and birds, which means that from these crocodile and birds are more similar, means the birds has been probably more similar to the crocodiles than to the lizard and snake. Similarly, in this clade too, if you see that the rodents and primates are more similar, although marsupials are very near to these ones, but not as similar as does not show similar homology as the rodent and primate shows to each other, right? So if we see the brief history of molecular phylogenetics, initially immunochemical studies used to be done that the cross reaction between the is stronger for the closely related organism, and this was Nuttall in 1902. Apes are closer related to human because their cross reactions are very strong. Then comes the protein sequencing method, electrophoresis, DNA hybridization, and PCR contribute to the boom in the molecular phylogeny. Right? In 1970 to present, discoveries using molecular phylogeny, endosymbiosis, divergence of phyla in kingdom by Wu's three kingdom classification in 1987, and many trees of life projects has been completed and underway. right? So what is the difference between molecular phylogeny and the morphology or physiology? See that Darwin has done the phylogenetic on the basis of morphology, because he used to or the physiology, he used to understand the environmental characteristics and the morphological characteristics of the different species, and then used to deduce the phylogeny. While the molecular data is different, it's not based on any appearance. It's depends on how this molecule, how the one DNA of one molecule, how the DNA of one species is similar to the DNA of another species. So if we see the differences, molecular data is strictly heritable entities, DNA, RNA, which are hereditary from heredity identities, which are heritable from one species to the another. And then the second, there is while for the morphology can be influenced by the environmental factors. Data is unambiguous. It is right because it is based on molecular characteristics. It's ambiguous and it can be changed according to the environmental factor and other factors. So you can, it's easily modifiable. Regular and predictable evolution, which means that it's predictable and it will not change with the time, right? The relationship between the two species will always remains the same and it will not change with the time. However, morphological or physiological, it is unpredictable evolution, because for same species when goes to another environment will have a different characteristics, so it becomes very unpredictable. Quantitative analysis on the basis of quantitative similarity, qualitative argumentation because it is based on the external appearance or the external characteristics. And it is ease of homology assessment because it gives a ease to the homology assessment between the two molecules, two species, while homology is difficult to access because of the morphology, morphological phylogenetics. Relationship of distance related organisms can be inferred by molecular data. Yes, because even though two species are uh, maybe look very different from each other or could be are distantly related, but they could be related to each other. Maybe because there are a number of different species in the between, but they you can always infer that even about the different about the distant relationship, while only the close relationship can be confidently inferred in the morphology. Once the species are too diverse, you cannot make out that what is the relationship or what is the tree for the true relationship. Abundant and easily generated with PCR and sequencing. That's the most important part, that with the help of PCR and sequencing, and as our sequencing techniques are increasing day by day, it is very easy to infer the phylogenetic with the help of molecular data. However, for the morphology, it can be, again, this is a problem because morphological can be very easily can be done for the higher organism, but when it comes to the bacteria, it is less visible and the morphological characters are less visible or they are lacking, and that's why you cannot deduce any phylogenetic relationship between the microorganism. Okay, now phylogenetic concept, interpreting the phylogeny. So this is kind of a tree which we get for the phylogeny. There are a number of different types of tree, but let's first read about this. So what is more important? This is the scale of time. As the time is passing, different species, initially there must be one ancestor, and this ancestor has been divided in two, because of the mutation in two different organisms. And then this one organism, again there was a mutation and divided in two, and then this organism, there was a mutation and becomes a C sequence, and then there was again the mutation and there is a sequence D and the sequence C. So what does this show? This shows that these branches are showing the number of transversion or the transition or the inversion mutations which has been taken from during the evolution time. One important thing is that physical position of this tree is not meaningful, because if this sequence C, D and E are here, and if I put this branch here, it is not going to make difference in any of the phylogenetic tree, which means swiveling can only be done at the nodes. At these nodes, I can invert this tree like this, and this sequence D and E will go this side and C will come this side. But this means that the tree is actually the same. It is only the method by it has been represented. So this tree and this tree are exactly same. Only thing is the method has been, so see, you can see that D and E is here and C is here. And here, sorry. And here C is here and D and E is here. So this shows that only the physical position of these branches has been changed and it is meaningless. Only that true evolution is shown with the help of the number of branches and how they has been developed. Okay. So the branching pattern of a tree is called as trees topology, and trees can be represented in several forms. Like this is a form of a rectangular cladogram or rectangular tree. This is a slanted cladogram. It's a slanting one. And there's a circular cladogram. Oh, I don't have a diagram here for the circular one, but it is just a circular one, which shows the relationship of one species to another in the form of a circle tree. I'll show you it in the coming slide. And then there is this tree. Tree terminology, and these sequences or the last species, last unit or last species or last taxon, it is known as operational taxonomic unit or OTU. So this is basically this one species is generally one OTU. So I'm saying it generally because not always. Maybe for one species you can have a number of operational taxonomic unit, and then that relationship will be shown between the same species as a subspecies. Okay. So operational taxonomic unit is in the phylogeny. It means that the number of sequences, similar sequences, are making one operational taxonomic unit. So exactly similar sequences from a sample, exactly similar sequences makes one operational taxonomic unit, right? And then, yeah. So these are internal nodes. These are terminal nodes, red one, and these are branches. So the tree can be, can be changed. It can be changed from in the position from at these internal nodes or the internodes. Root. These two species which are, or the operational taxonomic unit which are very near to each other, are known as systems. And this kind of tree, when there are three together, then it is known as polytomy, right? Then there's a rooted tree versus unrooted tree. Rooted tree means there is a common ancestor, but unrooted tree does not have any degree of kinship among taxa, but it only specifies the degree of kinship among taxa, but does not show any evolutionary path. So it's not showing any evolutionary path. Here we can understand that this is an evolutionary path, but here we don't know from where it is. The evaluation is taking place. Only it is showing the relationship between the different OTUs, right? And then there is a tree terminology between the scale tree and the unskilled tree. So scale trees are the trees where there's a scale is showing in the bottom of the tree, and this scale is actually showing the number of mutations in per 100 years or per 1000 years or per centimeters. Like suppose this scale is showing 10 changes, which means if this is 1 centimeter, 1 centimeter is representing 10 mutations. So if there is a 10 mutation, if this size is same as this size, it means from here to here, there must be 10 mutation which has been taken place. So these are known as skill tree. Branch length are proportional to the number of nucleotide amino acid chains that occurred on a branch. Usually a skill is included. Unskilled trees are the trees which just shows the relationship but does not show the how much amino acid or the nucleotides or the mutations has been taken place. Then monophyletic and periphyletic. Monophyletic is that in the same clade, in the same group, only the same kind of phylogeny, only the same kind of species are there. So here, like if you see, they are Saturnite and Martians. And while this is paraphyletic, means the species from the one clade is also appear in the another clade. So those are paraphyletic groups. Then the method of making phylogenetic trees. So there are three methods which we generally use. Are distance method, which is like neighbor joining method, then the maximum parsimony and the maximum likelihood. All of these are different algorithms which help in making the different kind of phylogenetic tree. All of these three are important depending on the type or depending on your requirement. These algorithms can be used. So the difference between these algorithms, like the distance, which is all we are also calling as a neighbor joining, and then the maximum parsimony and the maximum likelihood. So only uses pairwise distances. So you already know about pairwise distances, which we have read in the second unit of the course, our Coursera course. So where the initially the pair is made between the two species, and then these pairs are compared with each other. Only use the shared drive characteristics, use all data. Okay, so which means here the shared characteristics are used for making the tree, and here all that data is used for the tree. Minimize distance between the nearest neighbors. So it try to minimize the distance between the nearest neighbor, come together, and then it matches the others one. Minimize total distance and maximize tree likelihood given specific parameter values. Means it it tries to give the tree more recognition by using number of parameters. It is fast, can be done easily. It is slow, it takes more time, and it is when you do it with maximum likelihood, it is very slow. Easily trapped in local optima. So yeah, so locally it is very much optimized, but when you see in globally, it is not very good. Assumption fail when the evolution is rapid and highly dependent on assumed evolution model. This is for maximum likelihood. For the neighbor joining, good for generating tentative trees or choosing among multiple trees. Maximum parsimony, best option when tractable. Means when if the number of samples are less than 30, then this maximum parsimony is good. Good for very small data sets and for testing tree build using other method. So you understand the difference that because this is faster, this can be used for a larger data set. This can be used up to a data set when there are 30 taxons. Okay, and this can be used for generally we use for a smaller data set, because if you use it for a larger data set, it is actually slow and it becomes very, very slow to deduce the tree with the help of softwares. Right. So distance or the neighbor joining, using the sequence alignment, pairwise distances are calculated, creates a distance matrix. The phylogenetic tree is calculated by clustering algorithm using the distance matrix. So tree is calculated with clustering algorithms, and the example of clustering algorithms are UPGMA or the neighbor joining clustering. Okay, so this is what we know, right? Initially the pairwise alignment is done, and then with this pair, this third one is compared, and with this three, then the fourth one is compared. So this is a pairwise alignment. We have read about it in our course. Then the maximum parsimony, all possible trees are determined for each position of the sequence alignment. Each tree is given a score based on the number of evolutionary steps need to produce the set tree. The most parsimonious tree is the one that has the fewest evolution changes for all sequences to be derived from a common ancestor. Usually several equal parsimonious trees result from a single, right? So this is easy to understand. Just suppose this is a tree, and when one more is added, So if D is added here, if initially there are like the 3 species and you want to make the tree and add one more, so then the D comes. So D can be added here, here, and here. On the basis of similarity or on the basis of less distance or less mutation, each of these will be given a score. And out of this, which one has a less score will be used for the next addition of next species or the next section. And then this all will be given a score. And on the basis of this, when the least one will be used for the next one. So on this basis, this maximum parsimony goes. Maximum likelihood creates all possible trees like maximum parsimony method, but instead of retaining tree with shortest evolutionary steps, so it also makes the trees with like the maximum parsimony, but it does not retain the tree with the shortest evolutionary steps. Instead, employs the method of evolution whereby different rates of transition ratios can be used. Instead, what is that? It gives a penalty and a score for each transition and the transversion mutation. And on the basis of that score, it try to calculate the tree. And the lower is the score, the better is the tree for generated by maximum likelihood. Each tree generated is calculated for the probability that it reflects each position of the sequence data. Calculation is repeated for all nucleotide size. Finally, the tree with the best probability is shown as a maximum likelihood tree. Usually, only a single tree remains. It is more realistic tree estimation because it does not assume equal transition-transversion ratio for all branches. So you got it. So this algorithm is based on basically on the score of transition mutation, transversion mutation, and all those things, right? However, this maximum parsimony was based on the based on the score which is given when a new taxon is added to the tree. Okay, so when you make a tree, how much confident you are about the tree, that how confident you are about the tree, that the tree which has been deduced by all or any of these methods is right, and this can be used further, right? So for deducing this confidence, or for knowing this confidence, we use bootstrapping. So what is bootstrapping? Bootstrapping is actually computational method to estimate a confidence level of a certain phylogenetic tree. How it is done? It is done with the help of, suppose these are your samples, and you invert this tree from this. Now what the samples will then, with the samples, we'll make two more samples. This is a pseudo samples. And then again, we'll infer a tree. And then we'll again make a pseudo sample from this and again make a tree. And number of times these two species are coming together, that number will show that this tree is the possibility of these two species coming together is more than any other. Similar, like suppose if we make from this sample, if we make 100 different pseudo trees, and out of these 100 pseudo trees, when you make these 100 pseudo samples and then the pseudo trees and the trees, out of 100 times, 90 times if these two are coming together, we'll write here 90, which means that out of 190 times these two are together, which means we have a high confidence level that the rat and human are very similar to each other. If we just have 60 here, suppose, if we just have 60 here, which means that with these two, the relationship of turtle, only out of 100, only 60 times they came together, which means that we are not very confident about the relationship. So this is a computational method which can be done in each tree. So these are, suppose the bootstrap values are shown at the internodes. So if it is showing 100, means out of 100 times when we made the tree, these two are coming together. This is 65, means out of 100 times, only 65 times this clade was coming together with the turtle, right? So values are in percentages generally, and values more than 60 are generally shown. Okay, so these are some discoveries which has been made because of the phylogenetics, like the endosymbiosis, origin of mitochondria and chloroplast. So we can show that this is actually very similar to the alpha purple bacteria, mitochondria, and the chloroplast was very similar to the cyanobacteria. This can only be deduced with the help of phylogenetics, right? And the relationship between the different human races, that like the Indians are very similar to the Rwanda, Ethiopia, and South Africans, while the Italians, US, and UK are more similar. Uganda and Tanzania people are more similar. So these are the different things which shows that how different races are similar to each other, right? So relationship between the species HIV subtypes. Oh, sorry, this is for the HIV subtypes. So this is actually the HIV subtypes which are similar, which are found in the countries. All right, so there are some problems and errors in phylogenetic tree construction, inherent interest and weakness in different tree making methodology. More is better. Error inferred phylogeny may be caused by a small set of data or the limiting sampling. Unsuitable sequences, those undergoing rapid nucleotide changes or slow to zero changes over time, may skew the phylogenetic estimation. So that because the phylogenetic tree is mixed with the estimation that the number of mutations which are taking place with time are actually uniform. Mutation, duplication, inversion, insertion, deletion, etc. can give the inaccurate signals. Genomic hotspots, some regions of rapid evolutions are not easily detectable. Homoplasy, nucleotide changes that are similar but occurred independently in separate lineages are mistakenly assumed as inherent changes, right? So that is very important. Like there are two different species, but the mutation is in both of them are occurring at the same place at the same time. Maybe we can infer that as the same characteristic, but actually that is not the same characteristic. These are the separate lineages having the mutation at the same time at the same place, but are actually the different lineages. All right, so this is all about making the phylogenetic reconstruction. Now what we'll do, we'll make one phylogenetic tree, and then I'll help you in making this tree with the help of Mega software. All right, so just I have to change the computer. I have to take another one. Right. All right, so yeah, let's make this one. Okay, so we have to stop here, right? And then all right, okay. Share screen. All right. So can you see this? My guys. Okay. All right. So this is our, you know, the sequences which we have got from our Coursera alignment, like course in the week 4 left from the Coursera, and these were the sequences we used for doing the alignment. So I hope you all know about the alignment. So now we have done the alignment. So how to make the phylogenetic tree from this, right? So in this, if you can see, actually you'll, because only one screen can be shared, that's why I'm not able to show you the another thing. So anyway, so on this tree, on this when you open this alignment window, if you go on the top, there will be a tab which is called as data. In the data, when you go to phylogenetic analysis, okay, it's opening here. So phylogenetic analysis, you click phylogenetic analysis, and there's another window which will open here. Uh, Here, you cannot see this right now. Let me show. Sorry, I cannot show you that one. Yeah, it's here, I guess. Yeah. Another window. Yeah, so you can see this, and then construct neighbor joining tree. Okay. And then you say yes here, right? You cannot show this, you cannot see this window. Let me try if I can show you this. So once you do this phylogeny, this another window will open. Yeah, this one. This another window will open. Okay, here you see the test of phylogeny, bootstrap method 500. You do this and then click OK. Okay, and then your tree will come. So I'll show you the tree. This. So this is your tree which has been reduced from the mega. So it's very easy to do. You know how to do the alignment. You have done the alignment. Save your alignment file on the in the .fas format, and then again open the mega. Open that alignment. Go to file that data tab. Go to phylogenetics. Click on phylogenetics. It will open another window where there will be a phylogeny. Click on phylogeny. It will give you a construct phylogenetic tree with the help of neighbor joining. Click neighbor joining, and then you will have another window. When you open that, in that window, test of phylogeny, you set bootstrap values set as 500, and then click on OK, and it will give you the tree automatically. Okay, so this is all about the phylogenetic tree. I hope you understand this easily, and you can see that this is a phylogenetic tree, and the bootstrap values are also written. These are the percentage. Well, I will finish this right now, and then we'll start the interaction session in like next 10-15 minutes. So I'm stopping here it right now, and hope you will understand this very well. Bye.